Okay, we are on and back here. It's Wednesday, April 7th, finishing up 27, starting 28 today. Lipids, sugars 27. Homework 12 is due tonight for 27. The sugars should have everything already information-wise you need for that. Really, our last topic here in 27 is just DNA. We're going to go through the structure of DNA and how it operates. And then we'll also have, on Friday, we'll talk about metabolism, glycolysis, and citric acid cycle. And there we'll show you all the reactions, all the substrates and enzymes and everything. But again, we're just focusing on the organic reactions. So we're not having you memorize the overall schemes. There'll be quite a few reactions. I think there's 10 reactions in glycolysis, 8 reactions in the citric acid cycle. You just need to be able to look at an individual reaction and then predict what the product might be from that starting material. And the enzymes, we're not going to make you memorize the names, but we'll show you all of them. And on a quiz, I think coming up, the last quiz, what's the last quiz? Quiz 10, I think, due next week. You'll see you'll have some of the reactions you need to analyze. Put down the specific enzyme name or get the name kind of close, okay? Well, you have time, open book, open notes on the quiz, so you can get the exact name. But for test 4, don't feel like you have to have everything memorized, okay? We're just analyzing the reactions and relating them always to reactions we've already covered, okay? So you'll see the commonality, hopefully, between the reactions of metabolism and that. So test 4 is coming up next week, right? Monday and Tuesday, 26 to 28, and a few topics in metabolism, okay? But again, not memorizing the overall scheme there. The final is the ACS official final. It's a standardized test, and it's over 3 different days and by section. I sent out an email to everybody. Did you see that? And we've modified the content on Learning Suite so you see what that schedule is. And we can look on the overhead up here, Jesher, and see just real quick. Yeah, it's by your section. So you should know what section you're in here. We'll start Friday morning with sections 11 and 12 right here in this room, right? 7 a.m. Sorry about that. This is the official BYU schedule. This is out of my hands, and this is because it's the distancing requirements, okay? So you need to be able to be distanced there. And then 11 a.m. we do sections 1 through 4 in these specific rooms over in the Tanner building. Hopefully you know where that is. It's the business building, right off the hill here toward the north there. Gives you time to find that room. It's on the 2nd floor, and those rooms, you need to go to that room depending on what section you're in. And then Saturday, again, we start at 7 a.m. right here in this room with 5 and 6 sections. And then on Monday, look at that, we have 7 through 10 over in the MARB. That's the classroom building, right, to our northeast here. You should know where the MARB is. It's where all the biology and life science classes are, I think. Some math classes are taught over there too. But yeah, note your section, note your time and place. We can't accommodate changes because this is supposedly official to make sure you don't have conflicts between your other finals, hopefully. But yeah, it's out of my control there. I'll be there, and your TA or some of our TAs will be there to help with that test. Everything you need for the test will be provided except a pencil. All you need to bring is a pencil. You'll have scratch paper. Oh, you can also bring models if you want. We'll have a periodic table and scratch paper there for you to use. Okay, but it's closed notes, closed book, just like our regular midterm. Multiple choice, 70 questions. It is officially timed if we were to share the information, the results anonymously with the ACS. You have to finish within like an hour and a half, the 70 questions. But we'll give you more time if you need it. So don't feel like you have to finish within that time constraint. Most people finish well within the time, okay? But just pressure-wise, we're not holding you to that. Is that okay? Questions on anything? We can talk more about this later too as we get closer, but I like to show it there. All right, let's go back here to what we need to know. in chapter 27, and yeah, it is just DNA. And we left off with this, right? ATGC, the three bases that form the triplet codon code for individual amino acids going into proteins. So it's an amazing set of numbers, I think biology has, right? We've got what, the 20 amino acids that go in all the proteins. And then the coding molecule, right, in DNA is A, T, G, and C, right? And these are these nitrogen uh, heterocyclic bases, and they're at the anomeric positions here. In DNA, it's deoxy, okay? And we've got thymidine, cytidine, adenosine, guanosine, okay? That's the abbreviation form. You don't need to memorize those, but you should recognize some things here. We've got just a single ring here for the pyrimidines, they're called. And then the purines have two rings, okay? And what's arrayed here are what? Carbonyls and NHs. So these are potential what? Hydrogen bond acceptors, the lone pairs, and hydrogen bond donors. So that's kind of the key thing. And maybe from other classes you've learned the base pairing, and we'll get into that. What? A with T and G with C. And notice that's a purine with a pyrimidine, right? And it has the complementary hydrogen bonding pattern. 2 hydrogen bonds between thymidine and adenosine, and 3 actually between cytidine and guanine. Okay. Now you can't overemphasize the importance of DNA, of course. It's the master coding molecule in the nucleus of all cells. In us, it's what, 23 chromosomes. It's 3 billion base pairs. If you were to unwind that DNA and all the chromosomes and hold it out in extended position, it would be how long? About a meter long, okay, over 3 feet long. But those are all wound up in individual chromosomes, and they're actually wrapped around histones, which are other proteins, function kind of like spools. It's the most dense packing of information we know of. Far more information is packed in that small space than anything we know in a hard drive, any of the most advanced solid state drives, whatever, pale in comparison to the density of information that's found within an individual cell. Now, it's the coding molecule, but only about 5% of it actually codes or has gene information, right? Each gene is a coding region for a protein that's expressed by it. So let's go over here on the sideboard, do a little bit just structure-wise here. No electron pushing. Oh, I got a little electron pushing there, you'll see. But why are we talking about this in this chapter? Well, ribose, right? So here's the penta saccharide, the aldopentose, which is ribose. And I always remember it because they're all to the right, okay? And it is a D sugar, okay, but it's only 5 carbons compared to glucose, right? And if we were to cyclize here, and form the furanose form, the cyclic form, we have our anomeric carbon. And let's see, it's in the up position as I draw it here. So is that alpha or beta? Let's see. It's on the same side of the D position over here, the R. This edge is coming out at you. So don't forget about that. And you see the down here, well, that's the right position there. Okay, and here's our D thing with this last stereocenter up. And then this one is up relative to it. So would we call this alpha or beta? Yeah, it'd be beta because it's up there. Okay. Alpha is also there, but in nature, when we begin to make the nucleosides and put the bases on, and then the nucleotides with the phosphate at that position, it is the beta position. Of course, that's driven by enzymes now. This is spontaneous here. This is equilibrium, but then this is with enzymes putting this on here. And we've got a few modifications, right? We've got A, T, G, and C here for the bases for DNA. Notice if this is RNA, that's U. What does U stand for? You've probably learned that in other classes. We don't have thymidine T in RNA. It's what? U or uracil. Very good. And that's just lacking the one methyl group. Okay, so thymidine has that methyl group. Thymidine is actually made from uracil. There's a methylation process that puts that on. And so the bases are at the anomeric position. Now this is a full acetal, right? Whereas the first thing was the hemiacetal, okay? There's another modification here in DNA, it's deoxy. There's a process where you deoxygenate that position. And that's where the term comes from, deoxyribonucleic acid. By the way, where's the acid in DNA? Do you see a carboxylic acid? I like to ask biologists this question because they know all about DNA and everything and genes. I always ask them about the name. If it comes up, whatever. Where's the acid? Nucleic acid. Well, it's in the nucleus. Okay, what about the structure? Where's the acid? What would you say? Do you see an acid there? You actually see a conjugate base, don't you? And that's what the 5' position it's called. We have the phosphate on there. This is now called a nucleotide. And these phosphodiesters link the two sugars together. It's actually the conjugate base, phosphoric acid, okay? So it's similar to carboxylic acid. The pKa's are 2 and 4, so they're normally ionized at physiological pH, but that's where the acid name comes from. And then the hydroxy at the 3' position is where things are going to be linked with the diphosphate. But they start out on the 5' position, the primary position, okay? So know the basics of the structure and what that means, you know, going here to the acetal form of ribose. Okay, well, here are the key things that, you know, DNA starts out with. Replication, what does that refer to? That's copying DNA, right? So when cells replicate or divide, you have to double the DNA, right? You've heard about that in other classes. We'll show you a little bit about how that's done. The double helix will unwind, and one strand becomes a template for building the new strand, okay? Actually, the process here with DNA polymerase or RNA polymerase, when we do transcription, is very similar in that the one strand is a template for the new strand, okay? We'll show double-strand DNA and talk about it. They're complementary to each other. They're not identical. It's not the same strand, okay? So how does this work? Well, here's 3' to 5', just abbreviating the bases, okay? The thing that's similar, of course, is the ribophosphate, or diphosphate, as it's called. So here, the first one is complementary. This is our new strand here. Here's our template strand over here. And what? T, hydrogen bond, 2 of them with adenosine is brought in. And then we bring in what? C, cytidine with guanine with 3 hydrogen bonds. You'll see this in a second. And we make this bond right here. What? That's the phosphodiester linkage. So how does that work? Well, here's the 5' end. And it always synthesizes from the 5' end toward the 3' end. It's an anti-parallel arrangement. You probably heard about that too. Those kind of details for test 4, don't worry about it, but the chemistry and the functional groups is what I want you to focus on. So how do we make this bond right here? That's the question. Well, let's have a blow up of this structure. Here's adenosine, double hydrogen bond to thymidine, okay, you see that? And then how do we make this new bond here? Here's the next one that's a template for this. Now there's an enzyme that does this, it's called DNA polymerase. And that should make some sense. It's making a polymer, right? We've seen polymers all over the place. What's the polymer right now? Well, it's the deoxyribophosphodiester that's being put into place. And so 5' there, 3' here, and there's our nucleophile. And here's our electrophile. It's actually the triphosphate that's brought in. The building blocks for DNA and RNA are these triphosphate nucleotides, they're called, okay? So the base is already on there. That's part of the template action. And then at the 5' position, we have this triphosphate. And what? It's electrophilic right there. So bring your pair of electrons into the phosphate, push the electrons up, bring them back down, and kick off what? Diphosphate, or pyrophosphate, it's called, okay? That's our leaving group right here, okay? And we're just doing that phosphate transfer, okay? So DNA polymerase does this. And then you keep going. What would be the next one here? Well, it's another G. So we'd have another C there, another C there, A there, T there. And you can see that would go on there with that complementary templating idea. Does that make sense, at least conceptually? Okay. Questions on any of that? All right. Let's go back up here to the overhead gesture. And yeah, we're going to spend most of the time here looking at graphics. And all these graphics are on Learning Suite, okay? Under content, you can see the chapter overheads and chapter summaries. So look at these. If you want more details of this information, the supplementary McMurray book that is available over in the bookstore, or you can find wiki pages on each one of these topics, okay? But everything you need to know for test 4 is right here on these. And I'm just pointing out the structures and what you need to know. Again, you don't need to know memorize the bases, but the complementary hydrogen bonding patterns you do. Oh, yeah, I like to throw in the history. Why not? This is called the greatest discovery of all time. Okay. And I don't think that's an exaggeration. Why? Because DNA makes RNA, makes protein, they make everything else. So all living systems are tied together in this idea of this coding molecule, DNA. So I think it's impossible to exaggerate the importance of DNA. And the original structure, the double helical structure, was originally proposed by Watson and Crick in England there in 1953. The first paper here in Nature was just a one-page paper, but it correctly proposed the double helical structure, and it had the base pairing, and it provided a hint for how DNA would replicate, because they said it could unwind, and then one strand would be a template for the new strand. Okay, these things were known. They were known. Phosphates and ribose were involved, and the bases were involved, but nobody knew how they actually came together and formed a structure that would actually have this information that would be transmissible to the next generation. So it becomes a very important idea. It's based on a crystal structure, at least the preliminary one by Rosalind Franklin. Maybe you've heard about her. She was a graduate student at Imperial College. She had these crystal structures, or the diffraction patterns suggested that it had a helical structure to it, okay? Now, they didn't know the molecular details yet. That was Watson and Crick actually building models and thinking about the base pairing, okay? Now, they shared the Nobel Prize with Rosalind's mentor, Wilkins. However, Rosalind had passed away at that point. She had cervical cancer, very tragic story, or she would have shared the Nobel Prize, certainly, with these people. But anyway, she had a 12-mer of helical double-strand DNA she was able to get a good enough diffraction pattern that suggested the correct structure to Watson and Crick. Anyway, you can read about that. It's a great story. Here's what you need to know. The hydrogen bonding pattern between T and A. Here's the hydrogen bond acceptor, the lone pair, and then the amine coming off the donor. Now, this is an aromatic amine, like an aniline, so it's not protonated at physiological pH. Okay, so it's deprotonated there. That aromatic character there, the lone pair resonance there makes that not basic, so it's able to hydrogen bond there. And then we have a hydrogen bond donor here and an acceptor there off the purine ring of adenosine. So being able to match that up, you see the lone pairs there I've accentuated, and then the hydrogen bonding allow those to get close. Notice it's a pyrimidine with a purine, the single ring with the double ring. And then C and G, similar deal here, except now you have 3 hydrogen bonds. You have a hydrogen bond donor, acceptor, acceptor, which is complementary to the donor, donor, acceptor pattern there, okay? And that allows for the templating effect to create the new complementary strand upon replication. Question on that, okay? Don't feel like you you might want to practice drawing them out a couple times and make sure you lock that in, but 2 double bonds between A and T and 3 hydrogen bonds between G and C. And here's the double helix. It is anti-parallel, 3' to 5', and then 3' to 5' here. I'm only drawing a 4 there, but you see the hydrogen bonding there. You see the phosphodiester backbone. The negative charges are mitigated by magnesium ions on the side here. And the double helix that you're familiar with, it looks like a ladder or stair step in between the two. And that's the hydrogen bonding between the two, okay? There is a major groove, a wider side of it, and there's a minor groove side. And that's due to the topology of the DNA. Most of it is like this. this type of DNA, but there are other types here, B, alpha, and Z, which are kind of extended or smashed down a little bit. Most of it is this type of DNA, but don't worry about that. There are different structures of it. It's tightly wound and spooled up and crammed around the histones inside the nucleus, right? Okay, here's replication we just talked about, and it's a complicated process, actually. There are other enzymes involved. A helicase unwinds the double-strand DNA first, and then the DNA polymerase with the individual, triesters, right? The nucleotide triphosphates, always at the 5' position. That's where the electrophile is, the triphosphates at the 5' position. That's where the 3' nucleophile attaches there, and you can see that. They actually go in opposite directions. I'm not going into all that, but eventually you have the new daughter molecules, and so when the cells divide, you know, the process you've heard about in biology, you have to have the two completely newly formed chromosomes that are then pulled off and then sequestered in the new daughter cells. Okay. Speaking of nucleotides now, that's not the only ones. We've seen nucleotides here in a couple other contexts. So here's what the adenosine diphosphate linked to another ribose molecule with the nicotinamide group. And this pyridinium group is what? The oxidant in nature, right? So NADH is the product. When this thing oxidizes or accepts a hydride atom from a substrate, okay? So nucleotides are actually all over the place. Here's another reducing agent, FAD, or oxidizing agent, FAD, which can accept hydrogen and a pair of electrons to go to the reduced form, FADH2. Maybe you've heard about that. The two hydrogens go across the flavin ring there. And here's another very important one that we'll see in chapter 28, and this is acetyl coenzyme A. And here's another nucleotide over here, adenosine. That tends to be the common one. Phosphate, pantothenic acid, and then this thiol with an acyl group. This is the business end right here. This is the group that actually gets transferred and will make all the molecules we'll see in 28, the lipids. But this idea of nucleotides, you can kind of see how that was. And tide means you have the phosphate on the 5' position. Okay, nucleoside means it's just the sugar with the base. Okay, so the terms there, you know, that's not important either. Okay, a lot of drugs interact with DNA. So here's double-strand DNA bound to a protein, topoisomerase. This is just a snapshot of it with this anti-cancer drug, topotecan, right here. This is a relatively flat molecule that can actually intercalate, interact with the pi stacking interaction between the base pairs. So here you see a pyrimidine with a purine right there, G and C. And then you see the flat part of the molecule can actually bind in there. And this causes a single strand break in the DNA. So there's a lot known about how these relatively small molecules interact with the big molecule. This keeps the cell from dividing. It shuts down replication. And that's a key step needed in cancer to keep the cells dividing. So some of these cancer drugs especially interact with DNA. Here's one that actually forms a covalent interaction. Mitomycin has this 3-membered ring, this aziridine ring, that actually opens up, undergoes nucleophilic attack there. And then this amine can actually interact with another molecule there and form a cross-link with DNA. And that also interrupts things there. But mitomycin's another known drug there. And again, I'm just showing this for your information. Doxorubicin or adiormycin has been a very common anti-cancer drug through the years. And you see this flat region here. This also intercalates, interacts with the pi stacking, the aromatic flat regions of the DNA. There's actually 2 of them in this crystal structure with the double-strand DNA that you can see right there. Okay, let's get into this. Okay, so the next step, transcription and translation. So we went over replication. Transcription's very similar, although DNA is a template for making what? An RNA molecule. And the RNA strand it makes is what? Messenger RNA. And that has the code directly read off the gene region of DNA. So transcription is converting what? DNA into RNA. You should know that term. You've probably heard that in other classes. Okay, there's a lot of types of RNA though. This is ribonucleic acid now with what? A hydroxyl at the 2 position and what? Uracil in place of what? Thymidine, okay, the demethylated version. Okay, there's 3 forms of RNA to be aware of. rRNA, what does rRNA refer to here? What's the R there in rRNA refer to? Ribosomal, yeah. And this ribosome's a big molecule. It's kind of like glycogen. You can actually see it under a microscope. It's such a big molecule inside of cells. There's hundreds, thousands of these in each cell. This is the catalyst, actually, that puts together protein. So it's a catalyst. You could call it an enzyme, but it's not actually based on protein. It's a nucleic acid. It's an RNA molecule that's all folded up, and it has specific regions where it can bind different things. One of the things it binds here is mRNA or messenger RNA. Oh, it's drawn out there. We can help you out with that. So mRNA is the actual thing that's made from the coding region on DNA, okay? And it's a similar process like we said. The bases, the hydrogen bonding form that template and allow us to form what this thing, and we got the 5' to 3' ends, which is bound onto rRNA. And now what are these guys? tRNA. What does T stand for? tRNA, yet a 3rd form of RNA. What's the T stand for? Transfer RNA, okay? And what are we transferring here? We're transferring an amino acid, okay? So here we're merging the 2 chapters. Chapter 26, the sugar, the nucleic acid, with now the protein, right? The amino acids. And the protein is made by individual amino acids. Well, they're brought on to the RNA using a tRNA. There's 20 different ones. These tRNAs correspond to each individual amino acid. They're complex little molecules. Well, they're not really little. There's over 70, 80 or so RNA nucleotides that are together. Some regions are kind of double helical, some are kind of folded up here, some kind of have an open region. This is the anti-codon region, which corresponds to the individual specific amino acid right here. So these cartoon depictions are sometimes good. You got these loops and then the helical regions kind of on the side. There's the anti-codon. Here's where the amino acid is bonded covalently as an ester. And we'll see that. Okay, that ester is put on there by tRNA synthesis, specific for an individual amino acid. So this one here for lysine, this tRNA for lysine, you see has this UUU anticodon at the bottom here. UUU stands for what? 3 uracils in a row, right? And this one, what? Aspartic acid corresponds to CUA. That's the anticodon. And what's red here is what? The corresponding messenger RNA, the complementary thing. So UUU codes for what? AAA on the messenger RNA. So it's a triplet codon that's specific for each individual amino acid. And then what comes on to the ribosome, right? It forms that linkage, that amide linkage here, okay? And eventually the ester linkage between the tRNA is broken, and those tRNAs come off, and those can be recycled, those can be reused. And what builds up is the protein, right? The peptide chain. So here's the link between sugars and proteins, okay? But we at least want you to appreciate this. You don't need to memorize these codons or anything, but you should know what translation means. It's converting messenger RNA into protein. And how does it work? What are these esters and how does all this actually work? Well, we've got to blow this up and look at the atomic detail here. So here's the important step to see. Here's the activation making the tRNAs. That 3' position on the ribose is exposed. So it's acylated. You put an ester, which corresponds to what? The individual amino acid. Remember, there's 20 of them. So you need at least 20 specific tRNAs for each one, okay? It's a complicated process. Sorry about that. The initiation, there's some other factors that go into this to get the process started. The key thing here, maybe you've seen this in other classes, the first amino acid is always methionine. That's the start codon, AUG. And what do we mean by codon? That's the triplet base sequence there that's on the messenger RNA. The anticodons on the specific tRNA that allows that to be brought in and start the protein formation. And that's with methionine. Now, we're going to go from the N terminus toward the C terminus to do the synthesis, which is just the opposite of what we did with the solid phase DCC-based coupling with the protected amino acids. So nature brings them in individually as esters. And why esters? An ester bond is what? More reactive than an amide bond. Amide bonds, you see we're forming them down here, are more stable than the original ester. So there's our first one. Methionine with its tRNA is brought in. Our next one comes in here. There's multiple sites. It can accommodate multiple tRNAs and amino acids as they come in. So two are brought in close proximity. That's what enzymes do, proximity and geometry. That allows for the synthesis to occur. And there's a base on the rRNA, on the ribosome, that takes this proton off, allows the pair of electrons to attack the carbonyl of the ester, you see. You have your tetrahedral intermediate, like we've talked about. That collapses back down, and what? The tRNA comes off. It gets protonated. There's your individual tRNA right there. And you just form this amide bond. Yeah, we have to deprotonate that amine. That'll give us what? Our dipeptide. Two amino acids stuck together. Well, that kind of moves over. There's a translocation event. Don't worry about that. You'll learn all that in more detail in biochem. And then the next one comes in, and another amide formation. The ester is broken. That RNA comes off. Now we have a tripeptide, and so on up. So that just keeps polymerizing until what? We get to UGA on the messenger RNA. What's UGA? The stop codon. Maybe you've heard about that. That does not code for any amino acid. No tRNA has that code. It just stalls at that point and allows for a hydrolase to do what? To just hydrolyze that last ester. Okay, so it stalls and allows for this to hydrolyze. And then what? You have the intact chain that can fold up then into the correct protein structure. So here you can see how we go from the messenger RNA with the codon sequences. It's a triplet for each amino acid. So it has to be many bases long, right? 3 for each amino acid. And sometimes we have hundreds of amino acids in a protein. So you need 300 or 1,000 or so bases there in the messenger RNA, which allows for the big protein to fold there. Okay, so that's the termination step, as it's called. Now, these codons, maybe you've seen these before. There's a codon chart here. It's a degenerate code in that there are multiple sequences that code for individual amino acids. So 3 of them with the 4 bases, you can see that's what, 64 possibilities, 4 to the 3rd power, I think. And you can see here, here's how it's read. If you use the first one and use the second one, and then you have UCAG as the 3rd one, that codes for phenylalanine and leucine. Where's our start codon? Well, there it is. AUG is methionine. And that's the only one that codes for methionine. That's the universal start codon. We have a couple different stop codons, UAA, UAG, and UGA. So you can see some mutations there. Speaking of mutations, if you change one of the codons, either at the DNA or the RNA level, you can change the amino acid. So here's one we've talked about, glutamate at that 7 position on what? The beta chain of hemoglobin. If that's mutated or changed, we can get valine there instead. And that causes a sickle cell form of anemia. And so that would be GAA for glutamine. And if it's mutated to GUA, that can become valine. So mutations are seen and changes in proteins are seen at the codon region. So here's translation, here's the idea. And these triplet codons, you see there's methionine. Is this valine, GUU? Yeah, there it is, GUU for valine. ACA for what? Threonine, AAU for asparagine, and what? Glycine, GGG. Oh, why is there nothing for UAA? Well, that's a stop codon. There's no amino acid put on it. Don't write stop here on the side of your peptide if you do this on a quiz or a test. There's no amino acid stop, okay? It just causes the process to stall and allows for hydrolysis on that end ester, okay? Know what it means, AUU, UAA, sorry, there it is, end or stop. Yeah, and it's in red because there's no amino acid there, okay? I know what it means structurally, but that's translation. Questions on that? Anything on that? I don't want to go into a lot of detail there. You learn a lot more about it, of course, in other classes. Very important molecule. Though. Questions on any of that? All right, let's look at the overhead here for chapter 28, and we'll go on here to this topic, lipids. Yeah. So what do we have? Lipids? What are lipids, by the way? We've got some terminology here, right, where we need to figure out what these are. And let's see, why is this? It changes the size on me sometimes here. Let's get that off. Okay. Sorry, I've got to back up a little bit. Okay. There we go. Okay. Yeah, let's see. So we want to go through the chapter to show you exactly what we'll need to know here. Lipids are nonpolar small molecules. So we've seen the polymers now. We've seen proteins. We've seen sugars. These are the smaller molecules. They're very important molecules. They're energy molecules, fats. Why do we say they're nonpolar? Well, they're mainly hydrocarbons, okay? Then we've seen the saturated fatty acids, or the long-chain carboxylic acids, and the unsaturated long-chain ones. We've seen the acyl triglycerides already. And we've seen some molecules that result from arachidonic acid making things like prostaglandins. So we'll review some of that and some of the reactions here. The lipid bilayer membranes, the barriers between cells, are made up by these phosphatidylcholines, which have a glycerol alcohol there, and then 3 esters here for fats, but 2 esters here for the lipids in the membranes, and then the phosphate group here. So these are polar here, this part of the molecule, but this is nonpolar here. And that forms the basis for the lipid bilayer membrane, okay, which just come together based on the hydrophobic effect. We'll review that. We have some other little molecules here, which are hydrocarbons called terpenes, essential oil compounds or signaling molecules in plants and insects mainly. We'll also see these intermediates in higher animals to make steroids, okay? So we'll see some of this this chemistry here carrying over into these molecules. Cholesterol is a component of the membrane. About 20% of the membrane content is made up of cholesterol. It's keeping it fluid, the fluid mosaic model of the phosphatidylcholines along with cholesterol. Cholesterol is the precursor to all of the other hormones in the body, though. The 2 I want you to know is testosterone, which is an enone in the A ring, and estrogen, or estradiol, which is a phenol in the A ring, okay? Very different molecules, okay? This is the male characteristic molecule, testosterone. This is a female one, estrogen, okay? Then we'll do fatty acid synthesis. We'll show you how acyl-CoA can be synthesized and converted into the different fats and made into the precursors for the terpenes, okay? So we'll be clear on that and hopefully just go through the basics of it. Now, we'll start out with a very complicated looking scheme here. And this kind of ties in the 2 topics. We'll make pyruvate, which is shown right here, from glucose. So this is why we're talking about this. Glucose upon glycolysis will be converted into 2 molecules of this little guy here, pyruvate. Pyruvate's kind of a key molecule in that it's in the boundary between glycolysis and the citric acid cycle and making a lot of other molecules because it can be converted here into acetyl-CoA. Okay? And that's just with an acetate, a 2-carbon unit there. So let's see why pyruvate goes to acetyl-CoA. Let's go to the sideboard here, Jesher, and go through a couple things. And we're going to look at the cofactors actually first. And these cofactors involve the conversion of an alcohol here to a, and this is NADH. This is the one I wanted to look at first. The cofactors in a lot of reactions, this is NADH, so it's actually going to do a reduction of a carbonyl compound like this. So here we have acetaldehyde. How do we get to the reduced form of this? And it would be an alcohol, right? Okay. So, and then our byproduct here will be NAD+. How do we do this? Now, I'm starting out with one of these key reactions because it's so common to so many biochemical pathways. We've talked about NAD+ before. Here I'm going to show you the reduction here. And in the enzyme that does this, we've got a protonated base nearby. Okay. This is called a hydrogenase enzyme. And the cofactor here is bound also in the enzyme. And you can push electrons here and push this out with its pair of electrons and then grab the proton off there. So you can see this hydrogen right here ends up on the alcohol, reducing it from the aldehyde to the alcohol. And what does NADH end up as? Well, it ends up as NAD+. Okay. Now that's one reaction to keep track of. What if we can do the opposite process? So here we're doing the reverse. What if we need to oxidize things in nature? Well, the enzyme can do the same thing. Let's take the base here, grab the proton off. That'll liberate these electrons to go in. And look, this identified hydride here, and we can identify that as hydride. Now, it's not a free hydrogen negatively charged. It's on this molecule, but its proximity and geometry, the enzyme holding this in proximity with NAD+ now, and you see that goes in. You push the electrons back around and go back to NADH and the oxidized form of that. So that's one reaction to keep track of. And you'll see these used over and over again, okay? How about this pyruvate idea here? So if we can take pyruvate, which is this molecule here, and we'll get two of those from a molecule of glucose, okay? And we'll do glycolysis and show you how that's made. How is this converted into acetyl-CoA? And acetyl-CoA is a cofactor molecule. It has that big adenosine and a pantothenic acid and a primary sulfide and then an acetyl group, acetyl-CoA. How do we get that from pyruvate? This is a 3-carbon unit. How do we go down to the 2-carbon unit? Well, let's have CoA with the thiol on there, and our enzyme here is going to have a base, and that will allow that to attack here. And you'll actually have an intermediate that will look like what? This. And, well, yeah, let me draw out the carboxylate here. Okay. And what can occur now? Well, we've got this intermediate here that will look like that. And how do we get to the CoA molecule here? Well, we've got to be able to take this off. So how can we take this off? Well, if we push the electrons down here and push them here, look, we need to have NAD+ right here to what? You can see why these cofactors are useful now. We need to take off this molecule. We need to decarboxylate, right? And so how can we do that? Well, if we then push this pair of electrons in here, you see we can get the reduced form, NADH, and what? We can lose carbon dioxide. So that's what I wanted to point out right there to kind of get started on this topic. Now, acetyl-CoA. Let's look at this molecule right here. This molecule is very versatile. Why? Because it can undergo reactions with nucleophiles. And what? It is a sulfur ester, so that sulfide can come off, and we can transfer the acyl group to a nucleophile. Okay. What else could we do here? Well, instead, we could take this off with base and get what? Get an enolate form. So CoA can become what? A nucleophile, which would then undergo reactions with electrophiles. So we've got some versatility here within this molecule. Here it is functioning as what? A nucleophile. Here it is functioning as what? An electrophile. Okay. So we've got some versatility there with the thiol-CoA. Yeah, this is a big molecule here, and the 2 carbons come from pyruvate, but allow us to make a wide variety of different types of products. Let's just see some. I think we can do the fats today. And again, don't memorize this whole scheme here. This is very complicated using another cofactor, vitamin B1, thiamine, okay? But you can see how the pyruvate, we're adding a nucleophile here. It's different than the CoA one I just showed you here, but you see the different type of decarboxylation. Originally an aldehyde, and yeah, you can see the CoA going on there. Oh, and then hydride coming off. We did the decarboxylation coming off with a hydride. It's a little simpler to see. So it's a different type of process, but you see the NADH as the byproduct, and then there's the acetyl-CoA. Question? Oh, yeah, I left off a sulfur there, didn't I? Is that what the one you were? Yeah, someone pointed out I made a mistake on the board, and that's okay. Yeah, so sorry, Joshua. Yeah, the sulfur is still there. Okay, when you use it as a nucleophile there. Yeah, but very good. Hopefully it's correct in the rest of the overheads. We'll see. Sorry, come back up over here, Joshua. Yeah, so fatty acids, here's your acyl-CoA or your acyl carrier protein. It's also a sulfur ester, okay? This is a bigger protein molecule, and here's glycerol, which is the reduced form of the alditol version of glyceraldehyde. And you can put 3 of those on there, form the oxygen esters, the acyl triglyceride, from the thiol esters, okay? Saturated, you should know, are mainly what? Solids at room temperature. Unsaturated fats are mainly oils, liquids at room temperature. That has to do with the crystal packing effects, how they come together. Don't memorize, well, there might be a couple names here you should keep straight. Stearic acid is the C18 saturated fat, okay? Oleic acid is the monounsaturated. The membranes is a very similar process. You put 2 esters on there and then a phosphate. You actually use ATP to do that, put on a monophosphate. And then you put on the choline part here to get the phosphatidylcholine, it's called. And that forms this macromolecular structure we call what? The membrane within cells. So the little squiggles here, they look like little frogs, right? The chains here are the fatty acid long chains. And then the round part, it's the phosphate group, which is exposed to water, both outside and inside the cell. So that's just a cross section of a cell. There are other ones in the body, don't worry about those, sphingomyelin, cephalins, which are a little less common than lecithin. Now, arachidonic acid has 4 unsaturations. This is a 20 carbon polyunsaturated fat, we'd call it. Upon injury, it's converted into the prostaglandins. We've talked about that before. I'm not going to go through those details again. You should know prostaglandins always have these 5-membered rings, these cyclopentanes. And then the different patterns here affect how they act in different parts of the body, okay? But they're signaling molecules, localized hormones. Yeah, we actually showed you a little bit about how they're made upon injury, the oxygen coming in and forming the ring and doing the oxidation, but don't worry about that either, okay? just the basics of it. We did look at the NSAIDs drugs, the non-steroidal anti-inflammatory drugs, way back in chapter 19, I think it was. And these are carboxylic acids, aromatic ones, aspirin, and the other ones, Advil and ibuprofen, and some that you've seen before, naproxen. There's a whole bunch of these. And they're second generation or more recent anti-inflammatory, anti-pain drugs. It's also inhibit the synthesis of prostaglandin. And here it is bound inside the enzyme that makes prostaglandin from arachidonic acid. So it's a direct inhibition of that synthesis of the prostaglandins that's important. Now here, we're out of time. Here's the new thing, the fatty acid synthesis. We'll pick it up here on Friday. We'll be able to finish this. We'll do the terpenes and the steroids pretty quick. And we'll actually start metabolism on Friday. So be prepared for that. So we'll go a little bit into fatty acid synthesis. You'll see how the acyl-CoAs come together and polymerize to make the long chain fatty acids. Then we'll also get into cholesterol synthesis and a little bit on cholesterol and steroids. And again, there's only two steroids you need to know, and it's just an A-ring structure, right? There's an enone in testosterone and the phenol, the aromatic one, in estrogen, okay? And that should be enough to help you keep those straight. Okay, very good. Questions, come on down. If not, we'll see you on Friday. Very good. Okay.